All right, we have the first people come in. That's wonderful. We'll give it a few minutes. Right. I was just in Boston, and that was lovely. They have like all these, trees with their fall colors. It was really nice.
Oh yeah. Yours stay mostly green, I suppose.
Oh yeah, we have a selected trees but not. Yeah, it's mostly conifers and green trees.
Yeah.
That's good.
I was raised in Boston area in a town called Medford.
Oh, I see. Cool.
Yeah, and so they had a lot of the tree. I grew up with that tree changing in the fall. It's beautiful.
Oh yeah, wonderful. That's great. Very good. Yeah. All right. We're still before the hour, but we already have some participants. So welcome everybody. We'll give it 2-3 minutes. And yeah, Boston is a really beautiful city. It reminds me of Hamburg in Germany, like all the beautiful brick buildings, and it's a port city also. And so I feel like home.
Yeah, are you from the north of Germany?
Yeah. Near
Bremen,
Bremen,
Hamburg.
Bremen. Bremen, yeah.
so many great mass spectrometrists and proteomics people come out of Bremen. Yeah.
Yes. Yeah, it's funny to think. That's yeah. No, that's great. All right. Very good. Well, I love mass spectrometry is really a very global, as all science is, right? But I kind of always like the global aspect of science and instrumentation, and really while science goes beyond the borders, I feel like we see this in the kind of labs, right? The postdocs come from all over the place, and it's one part I really enjoy about science.
Yeah, global science, yeah.
That's right. Global questions, right? And yeah, it's great. Yeah, so the imaging I find particularly interesting because it gives us the spatial resolution, right? Which I think is something that I find just absolutely fascinating. And actually at our institute, we had like a little small talk from one of my postdocs who presented some work he's doing with Peggy, and it just it's so relatable for people, I feel, right? When they actually can see something other than a mass spectrum. So I feel there's some certain fascination in that. Neil and Kevin, have you seen this when you go and give talks? And I'm sure you get this feedback a lot, right? I mean, what do you think about that part?
Yeah, absolutely true. We're visually oriented beings, right? And so we see these images, and you can see some of the most incredible images, and it doesn't really even matter what it's telling us. The images of themselves are just spectacular. And to think about where we've come in the last 10 years, it's a long way. So it's great. Yeah.
Yeah, super nice. Yeah, great. No, that's so yeah, this is we're really excited. And so maybe I can start with a little introduction, and I'm sure we have still some more people come in, but we already have a really nice group here. So yeah, my name is Birgit Schilling, and I'm hosting this wonderful seminar today. And we have today Professor Kevin Shea and also Professor Neil Kelleher. And so for the format, hello. For the format today, we will actually start with Kevin, hear an exciting talk from him, and then have time for a few questions, and then we'll continue with Neil and have really a chance to see some exciting science today. And we'll have ample time for questions also at the end, or also for big picture questions you all may have. So take note of your questions, or you can put them in the chat, but we can also see if we can ask them live, but we can do the chat. But today, so I'm really honored that we're having this today. This is series number 2 of 3 that we will have these imaging courses before US HUPO in March in Chicago. And in Chicago, we'll actually have an in-person workshop on the first Sunday at starting the conference. So keep that in mind, maybe sign up for that if you're interested and intrigued by what you'll hear today. And this will be recorded also. So yeah, Kevin, maybe we'll start with you. I'm so honored that you have time to speak with us today. And I would just like to give a little background on your career path, which is just incredibly interesting. So Kevin received his Bachelor of Science in Chemistry from Muhlenberg College in Allentown, that's in Pennsylvania. And then you went for your PhD in analytical chemistry to Purdue University, and you worked with Graham Cooks. You did a postdoctoral training in Chicago. That's wow. So you got around quite a bit. And then you started your own lab at the Medical University of South Carolina in Charleston. And, you know, you've been there for 18 years. And then you moved in 2008 to the Mass Spec Center in Vanderbilt. And as I understand, you have actually also, you know, appointments in the chemistry and ophthalmology department. And really, that's where your real interest is, in the lens biochemistry, proteomics of aging, of ocular tissues. So I'm actually quite interested in that too. Exosome proteomics and imaging. And you lead the core laboratory of imaging at Vanderbilt. And you're actually a fellow of the American Association for the Advancement of Science, and also of the Association for Research in Vision and Ophthalmology. So I couldn't think of a better person to start our webinar today, Kevin. And thank you so much that you're here today. I'll give you the floor.
All right. Well, thank you so much, Birgit, for that wonderful introduction. I'm really happy to be here. Let me see if I can share the proper screen. I think that's wrong. Let me see this one. How does that look to you? Whoop, now I think you're muted. Does it look okay?
Oh, I think you just have to swap in the upper, it's still the preview now.
Okay. And now?
It takes a second, let's see.
Whoops, sorry. It was showing up correctly the first time on my screen, but maybe not yours.
You're still in the preview.
Yeah, let me swap one more time and see what happens. No, still in preview?
Yeah, it's still on preview.
Okay, let me stop sharing.
Right.
And just get this right. Sorry, everyone.
No worries. In our test this morning, it worked perfectly.
Yeah, so when I go to share screen, it gives me 4 separate options for my presentation. I'm not sure which one is the right one. Okay, let's try this.
Oh, this is perfect. Yeah, perfect. Excellent.
Okay, so here we go. Thank you. Thanks again for that kind introduction. I'm really pleased to be here to talk about spatial omics technologies and imaging mass spectrometry. So the topic I was asked to talk about is protein imaging, and I thought I would talk about imaging membrane proteins and modified proteins because it's something that that we don't talk about or really very much in the imaging mass spectrometry field. Okay, let's get this to advance. Okay, so what I'm going to tell you about today is just a brief introduction to protein imaging mass spectrometry, and then talk about modified proteins, membrane proteins, and then at the end finish with how we identify these signals that we're imaging, and then hopefully that's a reasonable segue into what Neil's going to talk about. So I'm sure many of you are familiar with imaging mass spectrometry, but just as an introduction, so we're all on the same page, the tissue that we're interested in imaging is sectioned. We can use fresh or fixed tissue, and I'll talk about the fixed tissue constraints in just a minute, but we take a tissue section, and I see a red laser pointer on there. Is that someone pointing?
Yes, we see. Yes.
Okay, that's not me, so I don't know where that's coming from.
Oh, that I don't know. I mean, you hopefully, maybe you could just ignore it. I'm not sure.
Okay, can you see my cursor?
I think
know
what is your cursor is.
Okay, hold on. I think I know what's going on here. Pointer options. Okay. Okay, here we go. So, after we take our tissue section, then we spray coat a matrix because we're using MALDI as our method of ionization. So we need a matrix on our tissue. We usually use the alpha cyano cinnamic acid matrix and for peptides, and cinnapic acid for intact proteins, but also some peptides as well. And once the tissue is spray coated, then we put the slide that contains the tissue and matrix coating into the mass spectrometer. We scan the laser across the tissue surface, and every place where the laser hits, we record a MALDI mass spectrum. So here would be a typical mass spectrum that we might expect. And then for any one of these signals, we can go into the software and ask where, for example, this signal might appear in the tissue. And if it's very intense, we color that pixel red. And if it's not so intense, we might color that pixel blue. And when you go across the entire tissue that way, you generate a series of ion images. These happen to be of brain that tell you where each particular mass to charge value is most intense in that tissue. So there are two approaches that we would use for imaging peptides or proteins. The first would be to look at the intact proteins. We typically wash with organic washes to remove lipids and some salts if we have a little bit of aqueous in there. And that is to improve our signal in the end for proteins and peptides. As I said, we apply our matrix and then acquire the image data. But in what we might call a bottom-up approach, we can actually, after washing the tissue, we can apply a matrix, an enzyme like trypsin, allow the enzyme to cleave the proteins of interest, then follow through with our matrix coding and imaging. And in this case, we're imaging peptides. And I'll show examples of each approach as we go through. So my laboratory, it has applied imaging mass spectrometry to a variety of ocular tissues. Shown here are just some sample images, and most of our work has been done on the lens of the eye, which you can see in this diagram, sits in the anterior part of the eye and focuses light onto the retina. And so this image here in the bottom left shows some signals for different proteins colored by different colors here. And you can see a range of different protein forms in the lens. And what's important for you to know is that the proteins that are in the very center of the lens are as old as you are. And the proteins that are on the outside edge of the lens or the cortex are relatively newly synthesized. So we have an age gradient of proteins from young in the outer cortex to, in my case, old in the very center of the lens or the nucleus. So we've been using the lens as a model system to study how proteins age for a long time. And while aging in the lens is not a huge deal, we can, if we get a cataract, for example, in older age, we can have surgery to remove that cataract and have a plastic lens inserted and problem solved. But as I said, we're using this as a model system. So some of the same modifications that we have found in the lens are occurring in other long-lived proteins in the body, for example, in the brain, which has some very, very interesting modifications going on there. But I don't have time to talk about that today. So what do the mass spectra look like from a lens? So if we cut a cow lens in this case, so either from a calf, a young cow, or a mature cow, you can see and apply a matrix. This is what you might see as a MALDI mass spectrum. And you might say, wow, that's not a very complex proteome that you're showing me. You're going to image what, maybe 4 or 6 signals. And we know there are thousands of proteins being expressed in the lens fiber cells. Well, what you have to realize is that the crystalline proteins, the so-called crystallins, are present at concentrations of about 200 to 300 mg per mil. So the proteome is dominated by these soluble crystalline proteins, and that's what we're seeing here. These are alpha A at 19.8 and alpha B crystalline at 20 kilodaltons. And this is doubly charged form. And in the young calf, young fiber cells, that's mostly what we see. And we've developed methods to look at other crystallines as well. And I'll talk a little bit later about how we dig more deeply into the proteome. But what's interesting about the lens is, as I mentioned, as we age, changes occur. So in the adult cortex, the relatively younger cells in the adult cow, we see a whole series of signals here in the lower mass region. And these are truncation products that occur spontaneously, spontaneous truncation or cleavage of the protein. And in the nucleus, where the oldest fiber cells and oldest proteins are, you can see more intense truncation products and less of the intact crystallines present around 19 kilodaltons. So what does it look like in human lenses? So we did an age study. And so what I'm showing you here are some images, ion images of a 7-year-old human lens. And these are alpha A crystalline products. So this is the intact full-length alpha A crystalline. And by the color scheme, you can see that it's most intense in the younger fiber cells in the cortex, but there is indeed signal all the way through to the center or nucleus of the lens. But there are other signals, smaller signals that represent truncation products that are present in the oldest fiber cells and in the 7-year-old lens. So even by the 7 years of age, we already see significant amount of protein degradation. When we get to about 29 years of age, you can start to see less of the intact alpha A crystalline, and even some of the larger truncation products are being truncated further in the older fiber cells, and these smaller truncation products accumulate. So by the time you're 50 years old, you can see there's no intact soluble alpha A crystalline in the center of the lens, and this large truncation product is only in the sort of inner cortex, and the center of the lens is filled with these small truncation products. There's many different ones, and we've just submitted a paper that characterizes all the small peptides that are present in older and cataractous human lenses. And it doesn't really change much beyond 50 years of age. We still see lots of small peptides in the center of the lens. Some of these have been shown to be pro-aggregatory, so maybe they're a cause of aggregation and light scattering as we age. I also wanted to talk about modified proteins besides truncation, and this is one of our nicest examples, and I'll just focus on the bottom row of images here. This happens to be a bovine lens, alpha A crystalline again, but what's really exciting about these images is that we're seeing in the lower row, it's alpha B crystalline. We see the intact, unmodified form, highly present in the outer cortex, and then it seems to decrease in intensity before it increases again, and in the center, it's being degraded. So what's happening in this inner cortical region where the signal goes down? Well, the predominant form of alpha B crystalline here is the phosphorylated form. So if we map an 80 Dalton higher signal, this is the phosphorylated alpha B crystalline that's dominant in the inner cortex of the bovine lens. And we even can map the doubly phosphorylated form in this same region. So when we overlay the unmodified and the phosphorylated form, the phosphorylated in green, we can now get an image or a picture of where the unmodified and phosphorylated form of this protein exists in the tissue. So something that's very exciting, especially if phosphorylation, for example, alters the function of the protein. So those were soluble crystalline proteins that I showed you images of. They're very abundant and we get good images from them. But my lab for a long time has been focused on membrane proteins, aquaporins or water channels in particular. So we very much wanted to image membrane proteins. And as you are probably well aware, these proteins are either embedded in the membrane in the plasma membrane or anchored to the plasma membrane. And so they're typically very hydrophobic. And so we had to develop a different sample preparation method. So we apply our tissue to the glass slides or the indium tin oxide coated slides using what we call methanol soft landing. It's just a thin layer of methanol that helps keep the tissue stuck onto the slide because we have to wash away the soluble crystallins that are highly concentrated, as I just showed you, in order to see the membrane proteins. So we wash the tissue with water. You can use formic acid, you can use urea, a variety of different approaches to remove those soluble proteins. And then we apply our matrix in a very high concentration of organic solvents. So 90% acetonitrile. We've also tried using hexafluoroisopropanol additive to our matrix solvent to solubilize those membrane proteins as much as possible. And the membrane protein, as I mentioned, we're interested in primarily is aquaporin 0. It spans the membrane 6 times, and it's very abundant in the lens. So it's a great protein to test our methodology. It makes up 50% of the lens membrane proteome, so highly abundant. So if you don't wash the tissue and you just apply cinepinic acid in a very highly concentrated acetonitrile, say 90% acetonitrile, you see this very ugly MALDI mass spectrum. There's a little bit of aquaporin 0. This is the full length form, 263 amino acids, and this large blobs of a signal or broad signal for crystallins. So not an ideal mass spectrum. But after, and that's the image, it's not a very good image. But after we wash the tissue, get rid of as many crystallins as we can, there's still a few crystallins remaining. Nevertheless, we get the most abundant signal being the full length aquaporin 0. And you can see the images look much, much better. So this is the full length aquaporin 0. And this would be a truncated form that's present in the older fiber cells of the center. So now we have a method to look at membrane proteins in the lens. So moving to human lens, aquaporin 0, what do we see? Well, we see the same sort of pattern of truncation that we saw for alpha crystallins. So the intact molecular weight is 28.2 kilodaltons. And in a 4-month-old lens, we see abundant, intact, unmodified aquaporin 0. By the time we're 21 years of age, though, most of the aquaporin 0 has been modified in some way. So the unmodified is only in the young fiber cells of the outer cortex. And the same pattern is seen as we age. So what's happening to aquaporin 0? Well, I hinted at truncation. So here are 3 major truncation products, 1 to 259, 1 to 246, and 1 to 243. And you can see that these colors, these signals are colored green, yellow, and cyan. They fill the center of the lens. So aquaporin 0 is being truncated, just like alpha crystalline and other crystallins are, as we age in a non-enzymatic fashion. So I told you I would get on to how we probe a little bit more deeply and look at other modifications. And that is by looking at tryptic peptides. So we apply trypsin and we image the peptides that represent the parent proteins in their location. So this allows us to look at other modifications, and I'll show you an example of that, other membrane proteins, and it allows us to image larger proteins. So the limit of MALDI imaging of proteins is roughly 60 kilodaltons. We've imaged larger proteins, but I would say the practical limit's around 60 kilodaltons. And we, of course, know there are many proteins that are larger. So how do you image those? Well, we image their peptides as surrogates for the parent protein. And this allows us to probe more deeply into the proteome and get the spatial information that we so much desire. So what do the tryptic spectra look like from tissue that's been treated with trypsin? And quite frankly, it's very rich. So this is an FTICR MALDI mass spectrum. I'm not sure exactly which tissue this is, but these are all tryptic peptides. And as you zoom in to this very small mass to charge region in the red box, you can see there are many, many signals and every one of these could be selected to produce an image. So many more than just a handful of images. We have hundreds, if not thousands, of signals that we can now image. So an example of imaging other membrane proteins in the lens, I'm going to show here where we were looking at the lens sutures. And the lens sutures are where the fiber cells come together at the poles, and the fiber cell tips form what we call these Y sutures in bovine lenses. And in the front or anterior of the eye, you have this suture. And also in the back of the eye, you have this Y suture as well. And these are cartoon diagrams, but if you get a cataract that's called a sutural cataract, these sutures become quite opaque, and you can see very beautifully this Y suture formation. And one of the questions we're asking in our lab is what proteins are specific to the suture tips where the fiber cells come together and form very close connections. And so when we image, we cut the tissue right at the anterior or posterior pole, we apply trypsin, and we image the membrane proteins. And again, we wash the tissue before applying trypsin, so we remove the crystallines. And we were very excited to see these Y-shaped suture patterns for several different proteins. The ones I'm showing you here are N-cadherin, shown by 3 different peptides. So one of the beauties of this approach is that if peptides from the same protein should have the same localization. And that's what we're seeing for N-cadherin, and ARVCF is another protein that's been shown previously to be important for suture formation in the lens. And the same pattern is seen in the posterior sutures. So now we have an idea of what proteins might be present in the anterior and posterior sutures. And very interestingly, other proteins are excluded from the sutures in these sections. So these are very well-known gap junction proteins called connexins. Connexin 50, shown with 3 peptides on the left, and connexin 46. You can see the Y shape is sort of a negative image because the connexins are present in the tissue that are not forming the suture tips. And the same can be seen for the anterior suture shown in the lower row. So we can probe more deeply into the membrane proteome. There are many other signals that I don't have time to show you today, and start to look at what's present in different regions of the lens. Now, getting back to aquaporin 0, I wanted to give an example of deamidation. So deamidation is a process where asparagine and glutamine residues lose their amide side chain, and they form aspartic acid and glutamic acid. So this is a shift of slightly less than 1 Dalton. So there's no way we're going to resolve this, mass resolve this at the intact protein level, especially with most time of flight instruments. So our approach in the lens tissue was to use the trypsin digest and image the C-terminal part of aquaporin 0. So residues 239 to 263, which contain several residues shown here that can be deamidated. Deamidation is the most prevalent post-translational modification in the lens. All the proteins of the lens have at least a single deamidation, or most likely many sites of deamidation. So the question here is, can we see deamidation? And if so, where in the lens is aquaporin 0 deamidated? And so if you look at the predicted masses for the undeamidated and the carbon-13 and 2-carbon-13 contributions, you can see that there's a significant overlap between the deamidated forms and the undeamidated isotopic distribution. So if you look at the 2-carbon-13 containing undeamidated form, these peptides differ by 0.02 Daltons. So we need to mass resolve them in order to image exactly the products that we want. And I wouldn't be showing you this if we weren't able to do that. So on an FTICR instrument, we can resolve, for example, the peptide with 2 deamidation sites and a single deamidation site with 1 carbon-13. And the images are shown here for a human lens. And so in the 4-month, we see predominantly a singly deamidated form. And by 21 years of age, we see singly deamidation in the young fiber cells, doubly deamidation in green in the inner cortex, and then a truncated deamidated form in the center of the lens. And this is also seen in an older lens, and in the center, the protein, the peptide is further modified. So now we can look at very small mass shifts and how these modified forms are distributed within the tissue. So at the very end, last few slides, I want to talk about how do we identify, how do we know these are the proteins and peptides that we're imaging? So we came up with a method to apply trypsin to our tissue, or you can do this at the intact protein level as well, and just take a small gel loading pipette tip loaded with acetonitrile formic acid. The extraction region's roughly about 2 square millimeters. And we take this extract and bomb load on a capillary column and do LC-MS-MS, either of the intact protein, so top-down proteomics, or of the peptides. And so in this case, for a lens, we extracted roughly this region. We wanted to know what this signal was here in the imaging mass spectrometry data. And just this 2 microliter extraction from a very small region of the lens gives us tremendous signal, get a beautiful mass spectrum, and we used electron transfer dissociation to get very fine sequence coverage of what turns out to be gamma D crystalline. So this method works beautifully. It's really quite fun to interpret these spectra and figure out what the signals are that you're imaging. And what about the peptide level? If you do the same thing, as you probably know, MALDI ionization and electrospray ionization don't give you the exact same peptides from a tryptic digest. They're 2 very different ionization processes. So one approach is to just look at what's the overlap between those 2 processes. The electrospray, of course, coming from LC-MS results, the MALDI from the imaging, so we only have accurate mass from MALDI. And we can get some identifications that way by direct matching. But we can also ask ourselves what proteins are identified this way and predict what other peptides might be present for those proteins. So we do an in silico digestion then of identified proteins, and we can identify roughly 2,000 peptides from this type of experiment. And in working with Gus Gray in Auckland, New Zealand, we came up with an automated way to process the data, that imaging mass spectrometry data and the LC-MS-MS data, and assign those signals, those peptide signals that we see from the tissue. So I don't want to go too long to cut into Neil's talk, so I'll end here with concluding that I hope you can agree that MALDI imaging mass spectrometry is a powerful way to do spatial omics. We can look at intact proteins, including membrane proteins and modified proteins. We can also image tryptic peptides that allow us to probe more deeply into the proteome. I showed you examples of modifications including truncation, phosphorylation, deamidation. And Birgitte asked me to talk a little bit about future, and I'll just very briefly touch on, I think spatial resolution is always an issue for us. We want to get to finer and finer laser spot sizes. Of course, our signal decreases as we go to small laser spot size. So we can do protein imaging and peptide imaging down to 10 to 15 micron right now. We can do lipid imaging down to 5 micron, and we'd like to go even lower. So that's a target. We'd also like to probe more deeply into the proteome. And so methods for imaging, preparing the tissue in different ways to get a broader coverage of the proteome is also a very important goal for this type of work. So I just want to acknowledge the folks that generated the data that I showed today, NDRI, where we got our tissue, and the folks who donated their tissues for research, funding by NIH, and support from the Mass Spec Research Center here at Vanderbilt. And thank you for attending. I'll be happy to answer a few questions before we move on to Neil.
Oh, thank you so much, Kevin. This was just very remarkable. I'm very interested to see what nice work you have done. So yes, I thank you so much. Please, anybody who has a question, you can post in the Q&A, or if you cannot find that, in the chat. But maybe, yeah, I can take a first question. So, Kevin, it's quite remarkable, I thought, how early you see these changes in the crystalline distribution, right? And so do you see any kind of associated vision performance aspects going along with that? Because I assume that typically would happen somewhat later, or were you surprised when you first saw this huge change initially?
Yeah, that's a very good question. And yes, we were surprised to see changes at such a young age. We have just recently completed a proteomics study, not imaging study, where we looked at just as deeply as we could into the lens proteome. And what was striking in those results is that there's a complete remodeling of the proteome at 50 years of age.
Mm.
And this is the age where we start to need reading glasses. This is a disease or a condition called presbyopia, where the lens can't focus on close objects, right? And now we have proteomics data that tells us that there is a significant change right around that age where this condition happens. So there's a lot of information, or there's a lot to learn, I think, from the protein changes. I do not think that we're really expecting a lot of change at such a young age, at like 7 years old. There's really not any indication that vision changes occur, significant vision changes occur at such a young age.
Mm-hmm. Yeah, interesting. And then also I was quite fascinated when you showed, as with age, then all of a sudden there was some localized accumulation of some of these crystalline truncations. So I'm actually interested, when you compare individual to individual, bovine or human, are there similarities in people, or is this a really unique personal signature?
So it's amazing how similar human lens aging is. But if you think about what's actually happening, it's non-enzymatic truncation. It's just protein instability, let's say, as a function of time. And so the most labile peptide bonds are cleaving as a function of age. So the similarity between humans is remarkable. We also see in some aging models of animal species the same processes occurring. Again, it comes down to protein instability, which peptides bonds are the weakest. And of course, our sequences the protein sequences are different. So the exact peptides are different, but the same processes are occurring.
All right. Well, Kevin, extremely fascinating. I mean, this was really inspiring, and we should talk some more, you and I, as we are very interested in aging. I think you have some really wonderful tools that you were able to show us today. So thank you, Kevin, so much, and good luck with your teaching. Thank you for stopping by at our webinar.
Thank you very much.
Great. All right. Yeah, so this was really interesting and fun. And yeah, now it is really my pleasure, Neil, to introduce you. And thank you so much for making the time to be with us here today. So Neil has quite a really impressive CV. And so you actually did your graduate work with Tag Beckley and Frank McLafferty at Cornell University in 1997. You then actually went to work with Christopher Walsh at Harvard Medical School. And you actually trained in high-performance mass spectrometry, and opened your independent laboratory at the University of Illinois in Urbana-Champaign. And then in 2010, you actually went to Northwestern University, and you, I think, became quite famous and known for your wonderful work on top-down proteomics. And it's quite nice to see your work that is both very technology oriented, but at the same time applying it to really cool biological projects. So I really like that. And you show it in over 300 publications. That's a lot. You also, in fact, provide this ProSite software. And I know that's a real important tool for many over 500 labs in the world. And you work a lot with FTMS and, have quite a large array of projects going over intact proteins, PTMs, chromatin, and cancer biology. So thank you again for being here today, Neil.
Sure, Birgit, thank you so much, and welcome everybody. Let me do the non-standard thing, and there's one question from Christina Clement, and I'm happy to have Kevin, who I think will be around for 15 more minutes.
Yeah, Kevin, are you still there? Yes.
Yes, I am.
Yes.
I was
Okay.
just about to type my answer, so not Niels.
Please take the airtime. Go ahead.
Okay, very briefly. The question was, what matrix would we use for plasma membrane lipid imaging or organelle imaging? And the answer to that is there's several that we use. Diaminonaphthalene works quite well. Dihydroxybenzoic acid works very well. We get beautiful lipid images that way. And the very title slide was an image of the anterior portion of the eye of lipids, so we can get down to 5-micron spatial resolution. We can't quite get to organelle resolution yet. That's a dream, a goal in the future. And the second question was for protein identification. I'm not sure I understand the question exactly, so if you could clarify in the chat, I'd be happy to answer, so as not to cut into Neil's time any further. But thanks for the question.
Yeah, thank you, and thanks, Kevin, for coming back on live. And
you thought you got off the hook, but we pulled you back.
We got you back. But
well, it's great. You know, Birgit, it's great to just be part of a community in a low stress, affirming environment, and to share some science today. So thank you for the invitation. And I've been inspired by Kevin's work for a long time, and finally was able to get into the game a little bit here in terms of imaging, which I agree, you were musing at the outset about the visual attractiveness of imaging, and then Kevin doubled down on that, and it's compositional proteomics. We've been working on that a long time, as you said, but now spatial proteomics, really important for 3 and 4D spatial biology to get better at its compositional analysis of proteomics. So that's what I'll be talking about. And Kevin showed it in examples of his top-down with ETD, their type fragmentation. So let me go into multiplexed proteoform imaging using individual INMS, and this is the work of Pei Su, a postdoc who actually came from Purdue, where some of the underlying technology was born there, insofar as small liquid interfaces to sample tissue. So let's get into it, and we've now got a good team working on this. But as I mentioned, compositional proteomics, we've been doing whole protein, and now we talk about whole proteoform analysis. And so it's clear to many in mass spectrometry, which is the audience I'll be talking to today, that when a modification arises on a protein, that if you weigh the whole thing, you'll see it. And so that modification naturally is PTMs, but also any source of variation. So isoform, coding polymorphism that changes the mass, these sort of things are captured. And so that's why we discover, characterize, and quantify proteoforms. This slide beautifully lays it out of the 3 main sources of variation we talk about all the time. Mutation or polymorphism creates a biallelic source of genetic variation. But then you have isoforms, short and long, and then you exponentially increase the number of proteoforms. As you combine all those sources of modification to protein or variation, you get this set of 8 proteoforms that Ying as described, and so you get all sorts of advantages. And I think even within the proteomics community, we don't pause to linger on these advantages, such as clarity about isoforms, such as allele-specific modification patterns, such as knowing the stoichiometry and capturing diverse modifications, and knowing, some people call it the occupancy. So here's a phosphorylation just on the short isoform, and it's present at maybe about 50-50. So these are all sorts of advantages of doing top-down, and you get them all. The disadvantage is that it's been hard to move from peptides to whole proteoforms, especially the big ones, and do that effectively. So in the last 3 or 4 years, if you haven't reconnected with this space, you should, because there's been a lot of, I'll call them nonlinear advances, or breakthroughs is not a misinterpretation or an overstatement, and I would call it a transformation towards next gen proteomics, doing it in a proteoform informed fashion. And so that paper I just left there, if you ever need a one-figure, one-page description of my basic intro here, that has it. So Birgit encouraged me to, lay out some basis for probably future Q&A, where we take out more broad questions than just proteoform imaging. So this has been my career track, long time now, 20 plus years. And we founded the consortium for top-down proteomics in 2013. And we continue to encourage more people to focus on proteoform measurement and directly asserting them, not inferring them solely. And then recently, as I mentioned, there's been a lot of increases in our ability to assert proteoforms, including within an imaging mode. And that's underpinned by this individual ion counting ability, which Thermo has just released this year as direct mass technology. So it has plenty of advantages that we'll get into. So protein imaging, it's been around a long time. Of course, at Vanderbilt, tremendous decades-long activity there, going back to the 90s, and also Jonathan Sweeler at U of I, from single neurons and tissue imaging. We want to get at their spatial distribution, of course, and do spatial biology, ultimately single cell biology, and to get at their functions. And so immunostaining does this a lot. There's just been a tremendous growth in the multiplexed antibody-based imaging technologies. I won't give you the menu of all of those, but that's been multiplexed out to, easily now 20, 40, 60 plex antibody-based imaging. And that's a whole area that's been really supported by the NIH Common Fund and the HubMap project, for example, the Human Biomolecular Atlas program. But even in all of that, it's protein is the unit of measurement, not the proteoform. So can we get a higher resolution type of proteomics? The answer is yes, and take up all the advantages which so many great people in mass spec imaging have done, the label-free nature of it, high feature capacity, and I bring in the proteoform ID, and then really explore at what level we can do this and at what spatial resolution. So we're, of course, building on the shoulders of others, and that naturally leads to nanodesi, MS, and a shrunk down version of this that involves a scanning approach. You can sort of see it here, and then scan. The problem is, how can you sample those droplets and obtain high capacity top down for ID? And you can see that's been a challenge in the field, long standing, in well over 10 years, and trypsinization has been one approach to really get at more proteins. Let's take another. And these are all papers here in the bottom that describe how people have approached this, either even in native mode nanodesi. That's particularly interesting, where whole complexes could come off the surface. People have tried with ion mobility to get better at separating out proteoforms, try different solvent extractions, change the buffers. So all this has been work, and you can see the papers are coming faster in this area. But what we did is bring this to the party, which is individual ion mass spectrometry, or I'll call it I2MS or I2MS. And it's an Orbitrap-based method. It uses charge detection technology, but brought into an Orbitrap. And so what we do, here's your afternoon paradox. It's multiplexed, but individual ion. So let me deconstruct that, where we can do 1,000 or many more than that ions at a time, but in each ion signal as it moves around the Orbitrap, we detect the image current directly for a single ion. And this right-hand plot over here is really the important part, where a single ion, if you integrate the signal that from a single frequency, and that's a single ion, and it works better for top-down and highly charged ions, 6+ and above. That slope that I'm showing here as a function of, say, a 2-second observation period for that single ion, that slope defines its charge. And that ability to assign the charge would then combine with the frequency, which we all know is mass to charge. That gives you the mass. So directly in the mass domain, in a fundamentally new readout of ion mass. So that's why Thermo calls it direct mass technology. And yes, I'm financially conflicted in that, FYI. But the point is that mass comes out of this approach, and it really is a very different world than Fourier transform mass spectrometry, where ensembles of ions and the constructive interference of all those frequencies gave rise to signal. Here, the signal is derived from each and every single ion, and the question is, can you combine that with nanodesi? Because one of the things we observed was that we dropped the concentration sensitivity, that the method of individual ion MS can work on much more dilute samples. So we talked about this years ago, and we were like, yeah, hey, well then the little droplets coming off of the surface of a tissue, fresh frozen, please, and yes, treated with just ethanol washes, would pair rather nicely with this detection technology, and has very significantly lowered our bias. So as we show in this paper, I'll reference it later, it has come out finally in the primary literature. We have a big increase in the size of proteins that can be detected in imaging mode. So yeah, up to 70 kilodalton is not at all overstretching this. And so we're doing exactly what imaging folks do, and that's just go through and do line scans, see what comes off the surface, integrate a lot of those single ions, generate proteoform masses and isotopic distributions for them, and then generate images. And so we even got help thanks to the HubMap consortium from the experts at Vanderbilt. So kidney tissue was the tissue that we were working on initially. And here's that paper that's come out in Science Advances. And so here were some of the bits that came out of that paper of scanning at a few microns a second. We can get a number of proteoform features that go up to 7 kilodaltons. And I just love that the data are really pretty clean, low numbers of artifactual adducts, et cetera. And what we see is detection with lower bias. And so the abundant proteoform has made itself known here. So we've got primary metabolite glycolysis as a classic example. These are obviously abundant or hyperabundant proteins, GAPDH. Well, there's GAPDH right there. And OXPHOS in mitochondria and the TCA cycle combined with glycolysis, these are really up in Go enrichments when you do all the analysis of this. And the identification of proteins can either use prior tissue proteoforms in a reference set of proteoforms and just know the masses, or we've also demonstrated off tissue MS. So, or tandem MS, off tissue top-down MS. And here that is, as an example, here's Vermentin at 53 kilodaltons. And we had previously shown that top-down using individual ion detection is advantaged, especially for big fragment ions. So high mass fragment ions, and that's depicted here in your lower right, along with the graphical fragment map in the far lower right. You can see, I mean, these are, look at that, it's a Y221 ion, a Y220, the Y218. You know, these are ions obtained from proteoforms that were directly off the surface, and that's a very significant advance. What else have we been able to show? You know, I mentioned isoform assignment. So this is glutathione S transferases. There are actually two different genes. One's GST-A1 and the other one's A2. These are two different isoforms. And dialing down even more, Pei and Jack and other people in the lab were able to show that this difference of 14 Daltons in these isotopic patterns, you know, when fragmented, allowed us to map what is a known coding polymorphism, dial it right in to this area here, this serine 111 to threonine 111. That's right here, and the tandem MS data type allowed us to really localize and pinpoint that, even right off tissue. Now you could say, well, what do the images look like for these? And they're not different. So the allele doesn't change the localization of this in kidney tissue, and I'll take you through more this kidney tissue in just a bit, including about a millimeter scale bar here, and we'll get into that in just a second. But that's the composition part that I wanted to focus on. So here's you know, some of the kidney maps we produced. Here's an autofluorescence image, which Jeff Spraggins and Richard and Kevin are well known to produce here. And this is vasculature here. Here's your scale bar. This is the outer medulla here of the kidney, and then the inner medulla, and then the cortex. So those are bog standard anatomy of the kidney. And now looking at some of the PIMS images here, we can look at an albumin channel here, distributed, somewhat enhanced by the inner medulla. And then there's integrated images on the right. Here's that GSTA I was focusing on. So glutathione S transferase, that one gene A1 in the cortex, and less so in the inner and outer medulla areas. Let's get into ApoA1, so a blood protein. We're getting an enrichment of blood proteins in the inner medulla. And this one's also, taglin is an interesting case where it's more enriched in the vasculature. There's a major vasculature flow right through here in this autofluorescence piece over here. So you can see that in our images here, where the taglin, for example, or smooth muscle cells are enriched right in that artery there. So that's an example of things we've done, and we've been at this for quite some time now in the interim. And so here's that same chunk of kidney tissue with the taglin enriched in the vasculature there, along with some of the other images what you saw before. And like Kevin, we were able to see some phosphorylations, not on aquaporin 0, but in this case on a heat shock protein beta, where we've got a phosphorylation, and there's some subtle differences in localization between these two proteoforms, as you can see in the green and the purple there. And Pei's gone on to, you know, really dial us into glomeruli here. So these are another part of anatomy of the kidney in just a small, thin section here, and able to look at the things enriched, proteins like vimentin, which are enriched in the glomeruli. And then in the vasculature, here's that taglin result as well when we really zoom in. So, you know, I think a Q&A session is probably better. I just wanted to whet the appetite there and focus on some of the takeaways, which are, hope that you can take away that, PIMS or proteoform imaging mass spectrometry, we can go readily up to 700 kilodalton. We've seen masses well above 100 kilodalton, but we want to just inch up on that as well as inch down on the spatial resolution. And we've got 21 proteoforms directly identified in the published report, and then 160 using that intact mass tag approach. That's the reference proteoforms that we look up by intact mass alone. And so, yeah, we've moved the needle up to more routine masses at higher masses, and then begun to really correlate this data with known anatomical structures and functional tissue units in the kidney. And we're moving on to other tissues, as you might imagine. And so the team really is the critical part here. So we have Pei Su, who is an underpinnings of this, along with Jared Gefader, Director of Instrumentation. Pei and Jack have been just crushing it there, really developing the full suite of tools needed to do this. And that includes the informatics. So we've got a bunch of folks active on that informatics. And then some of the I2MS, which has been commercialized by Mike Sanko's team, and some additional algorithm development. At Thermo has really broken this area open, and maybe quietly so in the pandemic. But really, it's been a team effort, and I'll stop there for the moment. But I got to thank Purdue and Vanderbilt. So some great measurement science coming out of the Midwest. There's that HubMap program I mentioned. This is a $600 million program now hitting its production phase for the next 4 years out of the NIH Common Fund. And then our National Proteomics Center, which Thermo has its industrial collaborator. So with that, Birgit, I'll just kind of turn it over and, let's have a Q&A.
Yes. Oh, wonderful. Thank you so much, Neil. This was very inspiring. And we actually already have a question for you. And the question is, it mentions excellent talk, which I can only underline. And it says, do you observe problems with lability of PTMs, or is your method particularly suitable for this?
Yeah, that's a great question. I think it's only going to preserve more than our grind and find methods. So we've done very preliminary comparisons of phosphorylated proteins and proteoforms, and they look somewhat the same. I don't think phosphorylation, when you do all the grind and find, that you're going to lose that. But now this person might be very sophisticated and be thinking about histidine phosphorylation, or things that if you do the pH change, that could really make more labile. I'd love to get into some of that, because it's all about preserving as much information as you can prior to the analysis. It's true of top-down. Now, if spatial top-down should really preserve even more, and I'd love to prove it more. But you've got to find, each labile PTM, there's gamma carboxylation. Certainly deamidation is something that happens artifactually in the workup of digestion and bottom up. So I think here deamidation might be able to be more faithfully studied, not because it's labile, but because you get introduced during sample workup with bottom up. So it's hard to be exact, but I just lay out 3 kinds of PTMs to give you some idea of what we're dealing with.
That's cool. Yeah, I was actually quite intrigued. Maybe I take the next question, and before there is another one already. So I was actually quite intrigued in your kidney work that you showed there, and you showed this protein CREAP, right, which you queue up. And so we actually observed that a lot in actually bone tissue and cartilage, and it's prone to senolytic treatment. So how, you mentioned also you're moving into other tissues, your technology, how do you envision to translate that to other tissues? Do you see a lot of challenges in that, or is it the form of matrix maybe that will have to be adjusted a little bit, or do you think it will be relatively seamless to move into other tissues?
Yeah, I think, you know, brain has been interesting because it's kind of squishy and kind of greasy. Yeah. So I think that's required maybe some changes, but I love your line of thinking around solvent changes, which I laid out all too quickly in the beginning, has a big source of bias that has been partially mapped out, but I think we could take advantage of those biases. The way that Kevin showed, you know, delipidation, and then he gets at the membrane proteins more directly, like aquaporin 0. I think that's a fertile ground for optimization. And but most of the tissues, you know, there's a scale for tissue squishiness. I'm being not scientifically rigorous, because there is a scale for that. And I think the firmer the tissue is, the better it is. And I was intrigued by Kevin's thought about the fixed tissue.
Yeah, right.
Because that, and you know, people say, well, oh, you can't do, several years ago, you can't do bottom up out of fixed FFPE. No, yeah, you can. And so bottom up might be top down might be in a similar situation. So
super cool.
So those are some of the considerations as we go to different tissues.
Super cool. Great. Thank you. And there's another question from Christina. She says, yeah, thank you for your amazing views on top-down proteoforms and imaging. She's wondering, can you use the data from top-down MS to perform a kind of LFQ label-free quantification of proteins of interest and measure the stoichiometry of some protein complexes in the mitochondria? That would be helpful from pharmacology when we measure the changes in the mitochondria damage due to some drug treatments.
Yeah, wow, there's so much biology in the mitochondria that's a great question. That the damage amount, especially for ROS and knowing the stoichiometry of the mods, especially those that are from an aggregation of misfiring of the mitochondrial, outer membrane proteins, should be accessible. And I'm using Carol Robinson's great work, and there was a science paper not long ago of using native mode, as Christina, you're suggesting. And also Helen Cooper is on top of this as well. She's moving in Birmingham, I believe she is, doing native imaging. So I think given how abundant they are, and this was denaturing mode, and using nanodesi, so the anonymous giraffe there, there's another question down below. But
I saw
it's nanodesi, that as you go into native mode, I think so many things will change. It's another source of buffer induced bias. But yeah, I think LFQ is going to be possible. You know, it's always that way, where first you show that something is measurable, and then right behind it, people want to now quantify. And so sure, and then you start off and think, oh, it's not going to be possible. There's this and that. There's all these sorts. But I think just separate precision from accuracy in your mind, and that we can quantify things with precision. And even if there's a source of bias, you can still do a lot of great biology.
Yeah, wonderful. Yeah, so you already answered the next question. That's great. But yeah, it's fascinating to see all the things that this opens up. And I mean, it's so interesting how things were different in the medulla and the cortex of the kidney, right? And yeah, so I mean, do you think if somebody, say, had like a succinylation of kidney proteins, they could be measured? Or do you think that then sometimes the stoichiometry of some very low abundant PTMs is too difficult? Or have you, do you have thoughts on that?
Yeah, you know, it's you do have to change thinking relative to bottom up. When you digest into peptides, you have an amazing ability to see modified peptides. I mean, just like 4, 5 logs
right.
of dynamic range. It's just so incredible. You could pull out this modified peptide, especially as the search algorithms, open search algorithms. But okay, so what's the relative abundance? What's the occupancy or stoichiometry of that mod? You don't know.
Right, that's right. Yeah.
And you can do a lot of experiments to know, but it's like that arc has been, it's just tough. But in top down, it's completely different. If we can get down to say 1% relative abundance, certainly 5%, if a PTM is present at 5% stoichiometry or above, we see it.
That's so cool. Yep.
And we know how much of the pool is it's there, how the stoichiometry. And then if we compare to another sample, and actually Kevin's talk really outlined that, where he showed just so clearly through aging, just there's the bird's eye view of what's happening.
Yeah.
So yeah, it's harder to get below 1%, I'll tell you that.
That's still really cool though. And actually what I love about this is you would kind of retain the crosstalk, right? If you have multiple modifications on one molecule. So, I mean, that's really the power of the top down, I think. And,
yeah, and all the sources of variation. So like maybe there's a phosphorylation on a truncated form of a brain of an eyeball protein. I'm trying to map it onto Kevin's talk. And that combination, you can never kind of predict where the biology has evolved to over 1 billion years. And so top down is highly connected to that proteoform analysis. Wherever the biology leads, you are sensitive to that, but in the 1% and above world of proteoforms.
You opened up a whole different world for me here, which is great. And so thank you so much, Neil. And this was such a wonderful talk. And, I would like to thank you and Kevin and all the participants. And I also really wanted to say, so Neil just mentioned earlier, he will be at US HUPO in Chicago, beginning of March. He will be there with 9 posters and lab members. So if anybody has any questions for Neil from now to March, come and see him and find him. And we have actually like a little flyer here. It's right at the heart of the river there. And yeah, well, Neil, thank you so much for coming today.
You bet. Chicago's right there. Yeah, that's great.
Exactly. It's around the corner. All
Thank
right.
you, Bert. Thank you, everybody, for coming today.
Yeah, thank you, everybody. And see you all in Chicago, I hope. All right. Bye-bye. See you.
Bye.
Peace.